Welcome to the Kratom Advocacy Podcast. My name is Lawrence, and today we have the pleasure and the privilege of sitting with Dr. Christopher McCurdy, who is an eminent pharmacological research scientist and a professor and associate dean, I believe, at the University of Florida. Dr. McCurdy, thank you so much for coming on our show.
Thank you, Lawrence, for inviting me. It's a pleasure to be here.
I know you've done hundreds of these, so I tried to keep the questions as unique as I could. I'm just going to jump right in because you're a much beloved man and many people ask questions, but also feel free to ramble as much as you want to. Question number one, do you think there's a good chance that habitual kratom use affects testosterone or the endocrine system in general?
That's a great question. We really have no idea yet. We've not done any work in animals to look at changes in hormone levels, whether it's male or female. We know that there is a historical sort of use as a performance enhancement for increasing libido, but I don't know if that's tied to with hormonal levels at all. I'm not aware of any other teams that are looking at hormone levels. We do see some sex differences in exposure in different species to the kratom alkaloids when they're ingested. But to what extent that is actually translatable to humans, we don't know yet either.
Well, I've read some reports online, of course, this is all anecdotal, of people taking traditional opiates, and of course I'm not comparing kratom to these as a medicine or treatment, but I've seen this said frequently that they seem to have permanent trouble with the heavy-duty opiates with their testosterone. Do you know why that is, and what, if you could extrapolate, what might be a connection if there is one, or not really?
I don't really know. I mean, that's an area that, in all honesty, I am not very up on. We don't do a lot of endocrine type work. We focus really more on the effects of opioids in terms of their pain relief, in terms of tolerance development, and side effects and sort of toxic issues. I started my career really in opioid industry, not in Mitragynine speciosa and kratom alkaloids, but really started in traditional opiate chemistry. And so I've always had an interest in understanding the pharmacology that's typically associated with those, and particularly that would cause dose limiting side effects. And so, expanding beyond really that focus of pain relief, and then sort of where does that translate into physical dependence and then addiction, and understanding those different effects. Never really have looked at other areas like, quite honestly, endocrine system, any metabolic disorders that could develop. We haven't explored any of those.
All right, then I'll move on to the next question. It's mentioned that kratom has over 50 alkaloids. Are there any unsung heroes of the alkaloid profile that are interesting to you? Trace alkaloids that have some potential medicinal value? We always hear about the Mitra and the 7-OH, but what others might be at play?
Yeah, I think it's going to be interesting to see how things play out. As we purified these and tested them, some for the very first time, screening against various nervous system targets, we've identified compounds that are very selective for serotonergic receptors. They could be involved in the mood elevation aspects. As we know, serotonin system has been targeted for decades for antidepressants and antipsychotics. So there's potential there for us. We're very interested in exploring some of those, and those are actually compounds that are present in just about all of the products that are marketed right now, and they are in high enough levels that they're probably playing into the effects overall of kratom, and that's theogynin and pananthine. So two different molecules that only differ by a double bond in a side chain on the molecule, and they have really identical pharmacologies. They still interact with opioid receptors, and they interact with adrenergic receptors as well. So they have this polypharmacology that a lot of drugs for mental illness and mental health have. Um, so those are exciting opportunities to us. There's a molecule, uh, I should say, they have their highest activity towards those serotonin receptors, though. And one that's interesting to us in particular is that they interact with 5-HT1A receptors, which are thought to be involved in reversal of opioid-induced respiratory depression. And so, um, there's been a lot of talk about the signaling pathways of mitragynine or mitragynine. It's kind of tomato tomato. We call it mitragynine because it comes from Mitragynine speciosa. But we truly believe there's a polypharmacology of all of these compounds playing together. And although in the literature there's a large discussion around mitragynine being a sort of signaling based molecule that decreases opioid side effects, and that may be part of the play. I think that it's the combination of, if you would, like an entourage effect of many compounds playing on different receptor systems that are sort of balancing out that whole experience. And when you start to remove these compounds and look at them individually, it's a very different situation because the pharmacologies, as you would imagine, completely change when you're focused on just one compound. And I always equate it to a complex symphony orchestra, because if you look at a symphony orchestra, you've got hundreds of instruments playing at once to produce an overall melody. Some things that you may not even be able to pick out in that song that are key if they were missing, and other things that you can obviously hear. But if you were to take any one of those pieces out and listen to it, you would be missing a tremendous amount as well. And so we think that there's this whole polypharmacological aspect of all of these, as you mentioned, 50, maybe more than that. We don't know. Consistently we can isolate in the 20s of alkaloids, a couple dozen alkaloids from no matter what plant material we have. And I know you probably want to talk about differences in plant material as well at some point, but we're pretty content. We know that some of the compounds that have been reported in the literature also could be artifacts of isolation processes. So when you're chemical manipulation to purify these compounds, the conditions can be pretty harsh as far as chemistry goes, and we can change molecules that may not exist naturally, but once we identify them, considered to be isolated from that plant material, even though they may have been chemically changed in the process. So we know that there's at least a couple dozen that are consistent that we generally see on a consistent basis. So far as the serotonin stuff goes, that's kind of interesting. We also know another compound, chorinanthidine, that basically is the same structure as mitragynine, but it's just missing a methoxy group. So a carbon with an oxygen is removed from the aromatic ring on the indole of the molecule. For those that understand the chemistry, that small change in the molecule completely diverts this molecule to having adrenergic activity. And adrenergic receptors are sort of where adrenaline interacts, or epinephrine. We can also think of it as the site of stimulants like the amphetamines. And they don't directly interact with adrenergic receptors, but they cause manipulations in dopamine, and other types of compounds that will interact with these receptor systems. Anyway, um, that kind of gives you that stimulant or energy type aspect of these plants, and so we think that there's some interest there in looking at these molecules that interact with the adrenergic system. That's also involved with blood pressure regulation and cardiovascular system, and in some aspects could be involved with um, sort of that uh, performance enhancement, if you will, the sort of increase of libido type claims that have been made. And so there's some compounds that fall into that pharmacological category, and those are really the three major pharmacological categories that we see. So serotonin, adrenergic, and opioid are the main. There's another compound that's actually a structural isomer of mitragynine called speciosialatin. It seems to have a little bit more activity at each of the receptor systems that mitragynine is, so it may be just a little bit more active type of mitragynine, but it's very, very similar in its receptor binding profiles. We know that it has very good analgesic activity, but we really haven't studied that compound um, to a large extent. And part of the problem with all of these alkaloids is mitragynine is the major one by far. It's the one to isolate. It's the easiest one to purify from a chemistry perspective, and that's been pretty evident within the literature because that's the most studied of all the alkaloids. Um, and after that, it starts to get difficult to isolate these compounds in a pure enough form that you can get in a large enough quantity so that you can really study the pharmacology associated with them. So yeah, there's a lot of other minor alkaloids that we've screened through many receptor systems that have potential to really drive different areas of research, whether it be in analgesics, whether it be in sort of mental health aspects, or whether it be in sort of energy inducing or cardiovascular system, lead molecules. We put out a manuscript. I don't know if it was a year ago or 2 years ago now, but we really call the Kratom alkaloids a blueprint for pharmaceutical design, because
Wow.
they're really there's really a a uniqueness to them, and being able to manipulate different pharmacological systems based on the templates that sort of nature has put out for us. And so there's there's a lot of excitement. I think I could probably work the rest of my career in diving into each of these individual ones to figure out what's even going on with them, let alone trying to think if we can inspire some analog or synthetic compound that would mimic their activity to become a pharmaceutical. And that's somewhat of the job that I do as a medicinal chemist. We are inspired by nature many times. There's at least 25% of the prescription drugs that are available that have been based on or are actually natural products. And so we obviously are interested in understanding the itself, but we're not interested in taking that natural product and creating a drug from it. We're more interested in what can this inspire us to create that would allow us to add something to society from the pharmaceutical side, leaving the natural product alone for its typical space where we believe it should be, which is available to people to help them.
If I may interrupt, I wanted to ask you. You mentioned earlier about the alkaloids being a kind of a symphonic thing with an entourage effect, kind of in the zeitgeist, especially in younger people. Mitragynine, 7-OH. These are the they're interested in isolating 7-OH. They're interested in taking extracts. And the other day I was at the store and I saw a man buying about $250 worth of these extracts. And I've never tried them before. I don't take kratom, but I said, all right, let me see what this is because I'm going to do it and see what happens kind of guy. Um, it was a powerful experience and it seemed almost engineered to produce feelings of pleasure. Um, and I very much, of course, I didn't take any more, but I very much wanted to. So it made me think there's kind of a war going on, not a war, but a sort of a debate going on right now about what will these isolates do for kratom's legal status and how might they affect society if the proliferation continues? It's kind of a vague question, but maybe you could weigh in on that.
Yeah, we've been very concerned about this as researchers, and we're, unfortunately, the street chemists are way ahead of us, natural products chemists, and the way that we can perform studies with all the regulation that we have to meet, whether it be in animals or in humans, which are even more rigorous to get approval for. Um, but we're still at the point of really trying to understand the leaf material itself. So, the leaf or the powdered materials that are out there. Also, some of the early extract products that have been out there. And up until really a few years ago, there wasn't much different available in the marketplace. But you point out something that has changed drastically in the last year and continues to evolve. It almost feels like on a weekly, and that is this sort of, if you will, arms race. Kind of what I was saying at is that in the last couple of years, there's really been this change in the products that are available in the marketplace, sort of an unofficial arms race between some of the vendors to put out more and more potent, or more and more concentrated extracts, which becomes of great concern to us that are trying to understand the health benefits and health risks of just the leaf material or some of the early extracts that were out in the market, like a 3x extract or something that's not anywhere near as concentrated as these products that are emerging. And it seems to be, something new every week. Not only do these concentrates keep increasing in their concentrations, we're starting to see isolates appear, particularly like the 7-hydroxy tablets. There's 7-hydroxy infused gummy bears now. There's all kinds of different products being put out there that we really have no idea around safety. And in all honesty, these are manipulated so far from what the leaf material is that they're really not kratom products anymore. And we've said, we published a paper not too long ago, my colleagues and myself, that all kratom or all kratom. It's another tomato thing. I've always called it kratom for years, for the last 20 years. So it's been kratom pretty much in the U.S. market. Kratom is what's pronounced in Southeast Asia. So I've always been sort of true to that.
Smart people can say that.
But we'll go with kratom,
Okay.
just so everybody on the same page. But we published a paper that says not all kratom is equal, and right now all of these products are considered kratom, from the leaf material, which is the closest to what is traditionally used, to these very manipulated, synthetically altered isolates like the 7-hydroxys. There's no way you can isolate as much from nature to put into as many units as are available in the marketplace. So these are synthetically made. Based on analysis that we've done of these products, we think they're taking Mitragynine isolates, and then they're oxidizing that through chemical reaction to produce the 7-hydroxy, It's not too different from how delta 8 THC is being produced, which is being made synthetically from CBD,
Does that
and
produce
you get.
contaminants? I'm sorry to interrupt you, but does that
I'm
produce
sorry.
contaminants? Does that produce contaminants in the process?
Yeah,
Okay.
absolutely, and that's what I was just getting ready to say is that delta 8 THC is made from CBD, and there's about 20 different compounds that are produced in that process that have never even seen an animal. Um, so we have no idea what the safety or efficacy or any type of toxicology associated with those. The same thing's happening in these 7-OH products. So 7-OH, we don't know anything really about that compound alone in humans. We know what it does alone in certain animals. We know that it is as pure an opioid compound as we've seen in uh, any of the alkaloids associated with kratom. Is probably one of the most selective opioid molecules I've ever seen in my life. And like I said, I basically have my expertise in the morphine type um, opioid alkaloids from the poppy, and 7-hydroxymitragynine or 7-hydroxymitragynine is more selective for opioid receptors than even the traditionally pharmaceutical opioids. And so, once we saw this 7-hydroxy, um, we've essentially called it like legal morphine. I mean, it's being sold with regulation. It's being sold as if it was kratom. It is, and it's dangerous. And although people may argue that they haven't, you know, a tremendous amount of deaths from this, or any deaths at this point, it's new into the marketplace. I know that sales are crazy, and I'm sure that what it's doing is causing some people to have euphoric experiences. Uh, may or may not be safer than traditional opiates in terms of respiratory depression. We have no idea if that's the case in humans, and so there's of taking any substance that's not even really been studied in animals. And then with the contaminants, along for the ride, there's really no idea of what all is happening.
Okay, and you said that no one's died yet from this, but you die every time you spend $250 at the convenience store on isolates, I'd imagine.
I guess. I mean, it depends. I say that no one's died, but I don't know that for sure. Um, and we know that there are deaths that have been associated with kratom products, and so whether they were causative or not is not uh, sort of a question that we can even answer yet. But the fact that they are associated with it, with someone who's died, always a possibility that any substance causes someone to die, unfortunately. And there's not tremendous amount of people dying from kratom products relative to the number that are supposedly using them. It really comes out to be somewhat, not on the dangerous side of products that are available in the marketplace. Um, Certainly, if you compare it to something like alcohol, it's much more widely used, but there's a lot of documented deaths associated to that. So it's a big debatable question. We don't know what toxic levels are, but I always like to stress that, as with any substance, even water, you can die from ingesting too much water. So there's water intoxication, people that die every year from drinking too much water too fast. And this is certainly the case. And Paracelsus said hundreds of ago um, the poison is in the dose, and as people start to escalate usage of substances, particularly these concentrated extracts or isolates, where we have no idea um, what what their what their levels are going to be when they ingest these substances, compared to something that's been traditionally used, which is just the leaf material in Southeast Asia for centuries. I kind of equate it to what we love to do in America, and that's supersize everything. And we've taken something that's been out there in nature for years, and we've sort of concentrated it up and tried to make it into something that it is absolutely not meant to be.
All right. So can you maybe that's can you talk about? I guess you could start by explaining about how kratom has agonist and antagonist opiate effects, and how maybe some dangers could be posed. Are you removing that modulating influence when I'm assuming when you just do an extract or process an isolate? Because if you take higher and higher doses of kratom, there are opiate antagonist effects, correct? But if you keep taking higher doses of 7-OH, would that same regulatory effect come into play, or no? Or
Yeah, unfortunately, there are questions we can't answer yet. There's just very little data known about the toxicities, even in animals. It's difficult for us in a regulated environment of research and animal care to perform toxicity studies these days. So our governing board in the university systems is what's called IACUC, or the Animal Care and Usage Committee. That that's very strict on what we do with animals, how we you know treat these animals, what kind of exposure to substances we give them, and we're very cautious of those animals into any harm. And so in the past decades, and I would say 20, 30 years ago plus, it was not uncommon at all to conduct what's called a lethal dose 50 study. So what's the lethal dose in 50% of the animals? What's a lethal dose in 90% of the animals? And so from this, we can start to understand where a substance will start to kill people, at what concentrations it will kill them. Now we have to do what are called maximal tolerated doses. And so we give an animal a dose up to a range where we start to see sort of severe side effect, whether that seizure, cardiovascular event, something that's we can medically treat that animal to bring it back to normal, but you don't want to have an endpoint of death. And so unfortunately, we can't do those types of studies very easily anymore, if at all, to understand where toxic levels are going to be in animals. And you know, we are collecting data in humans, unfortunately, that have passed from ingesting too much um, substance, and whether or not it was in combination with other substances adds a whole nother layer of complexity. But we're starting to understand sort of where a range of exposure can be, and that's just for leaf material. We have no idea in these hits. We have no idea in these concentrated products. We do know that products or isolate products, dramatically increase the risk of getting some harmful event happening, because it's just like anything. If you look at alcohol, and if we call alcohol a general category, you're looking at everything from a light beer to something like Everclear that's 95% ethanol. And of course, if you were to do um, an ounce for ounce comparison of a light beer, which may be 3.5% alcohol in that ounce, versus something that's 95% alcohol in that ounce, you know that if you consume multiple of each end of the spectrum, you're going to get to a much more dangerous space with that very high concentrated alcohol product versus a very diluted product. And so our point from the research community is we have this very big increase in risk of developing harm for a product just because it's been concentrated so much.
It's a shame. It sounds like all of these ethical concerns make it difficult to compete with, as you said. I see commonalities in a lot of sectors of society. Like just a gun dude was saying before. Well, these garage inventors are getting around the regulatory bodies, and they can work faster than we can. So, I mean, I hope that things turn out well for you and your people. It sounds like you're doing the right thing, but the right thing is often seems cumbersome.
We're
Um,
trying, and unfortunately, it's
mhm.
just that you know we be able to keep up with public usage. Science is a pretty exact art, and it's pretty slow to get that exact. And unfortunately, we're following, and we're behind the eight ball most of the time, trying to keep up with what trends are happening or what we're seeing. And many times we're finding out about these isolate products because we actually see them in a gas station when we go to get gas, or we see them somewhere and say, "Oh my goodness, where is this?" And immediately purchase it and take it back to the lab to try to figure out what's happening. So um, it's bad when you have to do research in a reactive way instead of a proactive way.
That's pretty cool. So you heard it here first that sometimes lab and lab techs they get their stuff from the gas stations too, except they're not using it the same way. All right. Um,
Yeah.
I would like to talk next about one commenter wants you to discuss how the kratom you use is selected for analysis, um, and if you could talk about ben twangy or fermented leaf and how that process affects or
uh,
does not affect the alkaloids.
yeah. So let me start with the first question, and then we'll get to the fermented stuff, which is kind of interesting. Um, Certainly, for leaf analysis, we have done a tremendous amount of research, from harvestation of fresh leaf material in Thailand, harvesting fresh leaf material in Malaysia, and harvesting material in Indonesia. And so we want to understand as close to the traditional usage as we can, and then understand how the chemistry of that material may change as it travels halfway across the world as a dried leaf material. So we've really looked at freshly harvested leaf, which is all the traditional use is either freshly plucked and chewed leaf, as they do mainly in Thailand. And in Malaysia, it's mainly freshly picked and brewed into a decoction or a tea for several hours. And so there's a difference, because anytime you pick a leaf off of any plant, that leaf starts to die because it's no longer getting its nutrients and support and all the other things that it has when it's attached to the living plant system. And with that starts to come chemical changes. We can see that leaf obviously lose moisture. It's going to be exposed more to oxygen, be exposed more to other elements that won't allow it to respirate or dry off or cool off anymore. So it's going to be heated. And so the materials that come to the United States, and we've gotten leaf material dried from all three countries in Southeast Asia that are mainly producing this as well. And then we've done a great partnership with many of the vendors in the United States that are willing to provide us with materials, raw materials, for research purposes. And so we get materials from various um, manufacturers, if you will, that package into capsules or into extracts. We get extracted powder materials to study. We've been very fortunate with um, a large amount of vendors that are really interested in understanding the science, really understanding the benefits that um, kratom can have for people, and wanting to understand where do we see harmful effects starting to develop. And so they've provided to us over the years, and it's been 20 years almost that we've been studying this now. We've just been very fortunate to have that generosity with no strings attached to those donations, and we've been fortunate, as I say. Not only that, we've been provided with a lot of products by uh, certain vendors that are concerned about a product that's on the market. So they'll send us those products and say, can you look at this? Can you see if there's any adulteration in it, or can you see if this is a legit product? And so we get products from all kinds of aspects, I guess, is my bottom line. We also, as I mentioned, we'll go out, and if we see something new that we've never seen or heard of, we'll purchase it with our own money and bring it into the lab and see what can we learn from this, and what can we understand this product is doing to the ever evolving sort of marketplace that's there right now. So we have a lot of our own ideas of what we want to explore, but we also are very much driven by what's happening in the market, and so we'll just get what may be the next thing, right? That people are starting to gravitate toward, and we want to investigate that and study that and put it into animals and see what kind of effects also that is producing. We will from time to time do some surveys, and we've had some grant funding and collaboration. Dr. Kirsten Smith at Johns Hopkins, where we've studied um, actual samples of individuals kratom that they're using. They'll send us a dose, and we can study that and see how that correlates to their sort of effect, or the what we call their pharmacodynamic, or their feel, right? What are they getting out of that product or that dose? And really, when we started to do some of those types of studies where individuals were sending us materials, we first really learned about the fermented product. Um, and we started to do some analysis of fermented products, and we haven't fully unblinded all of the materials that we have, but we do know that um, we had a couple of samples sent to us that weren't at all kratom. We didn't go back and look and see what it was. I mean, somebody could have just been playing the joke and sent us parsley as well or something. Just kind of like, haha, we'll see how good these guys are. But we don't know if that's truly the case. But the fermented material is definitely different. I can't say in what all is in there. We do see some of the alkaloids that you would see in unfermented leaf material, but there's other things there, and quite frankly, we haven't had enough of it and enough funding to really pursue a deep dive into those fermented products. And so they're definitely, you know, for lack of a better word, contaminated with other things that we have no idea what they are. But you would expect that in a compound that's been sort of manipulated or changed. And what those are, right? I can't even speculate at this point.
Okay, thank you. And another viewer said that he thinks that all these lab test certification QR codes on the packages are BS, and they don't really mean much. Could you explain, is there any validity to that claim, how that varies? And something else I forgot, but yeah, we could start there.
Yeah, so I think it's really I think it's a really great step in the marketplace that vendors are actually putting QR codes on so you can tie back to a certificate of analysis for that particular lot number or batch number. And some of those are legit. Some of those may not be legit. I do know of a couple companies where it is actually the exact of analysis for that exact batch. Um, and it's great that they're doing this. One of the big concerns that's come up in the industry is the fact that a company can send off their bulk material to 3 different labs and they'll come back with 3 different results. So there's no real consistency in the industry for testing standards. It's not like we go to the National Institutes of Standards and Technology or NIST and say, hey, can you provide us with the Kratom alkaloid panel so that we can look at exactly what the standard should be. We have had discussions with NIST actually from our lab to try and produce some of those for the industry. But even if you have those tools available to you and those certified standards, we're dealing with different methodologies that are being utilized to analyze these alkaloids. So the gold standard for us is what we call UPLC MSMS, which is ultra high performance liquid chromatography with 2 mass spectrometers attached to it. So we call it tandem mass spectrometry. And what that allows us to do is perform very good separations of all the alkaloids. And then we can actually take the exact masses of those alkaloids and we can prove that those compounds are what they're supposed to be. We can fingerprint what the profile should look like, and so we can start to determine we can actually look at, and I know you had sent me some things about strain oil, but we've been able to identify essentially what would be like a material from a bumblebee strain versus something that would be from a more traditional red or green strain. And it basically comes down to the presence of mitragynine or not mitragynine. Bumblebee strains really don't have much mitragynine in them at all. They have a compound called mitraphyline, which we don't know very much about the pharmacology of, but that's the predominant compound in those. And so either what we see in an analysis, in a sort of a fingerprint analysis, if we see high amounts of mitraphyline, these are coming from the more bumblebee-like type strains, versus if we see high amounts of mitragynine or mitragynine, we know that that's coming from a more traditional sort of strain of Mitragynous speciosa. We've gotten into strain involvement with our horticulture colleagues at the University of Florida, and we've started to do some simple things, what are called DNA barcoding, that allow us to look at DNA base pair interactions to authenticate or sort of do a genetic analysis to say that, yes, this is Mitragynous speciosa. And we're doing full genome analysis now on several of the Mitragynous species. So there's 10 different Mitragynous species, speciosa being the one that everyone is sort of familiar with in terms of kratom. Although many people will say that any Mitragynous species is involved in kratom production. And so we've heard about dinosaur kratom, maybe not as popular anymore, but it was out there. It's because the leaves are huge. It's actually from a species called Mitragynous hirsuta. And Mitragynine hirsuta doesn't produce very much Mitragynine at all. It's much more closer to the bumblebee strains, where it's producing large amounts of mitrophyllin. And so we've started to look at this in much more detail at a genetic level within the plants. And the other thing that we know is there's a species called Mitragynine diversifolia. Mitragynine diversifolia again sort of pairs a little more closer to the bumblebee strain. And what we're noticing is at some point in time, somewhere, and we've seen it on living trees, individual living trees in Malaysia. We believe that Mitragynine diversifolia and Mitragynine speciosa have crossed in nature, and this creates a unique sort of alkaloid profile itself as well. And so we did GPS harvesting on specific trees at a plantation in Malaysia with our colleagues at the University of Science Malaysia. And what we were able to notice is that there was high Mitragynine producing trees next to trees that were hardly producing any. And so, we really want to do genetic analyses on those specific trees, go back and look at those. But we've been doing a lot of genetic analysis on trees that we have at the University of Florida. And this is where we're seeing a lot of these differences, and then being able to take those live specimens that we have and then compare them back to a lot of the powdered leaf materials that we have. We can start to cross-reference and fingerprint what we think certain products are originating from, if you will.
Could you geographically trace these contaminants to certain clusters of areas? Do such clusters exist now? Hey, we've got lots of contaminants coming from here, or is that just impossible?
Yeah, it's really impossible. And knowing the fact that a traditional stand of trees in Malaysia may have this variation within it. The farmers are harvesting leaves, and then they'll give them to a brewer or a producer, if you will, and they don't pay any attention to the chemistry. They don't have any idea what's going on with the chemistry. It's a natural product. They brew it into the tea. And individuals will consume the tea. It's not too different from how we coffee in this country. Nobody really cares or examines what are all the different alkaloids that are in my coffee. Of course, we care about the caffeine, right? But the caffeine levels, even within beans that are harvested in the same product, and now they're very consistent in terms of their roasting and the way that they produce their coffees per company, much more consistent than anything in the Kratom business. But you're still going to have an individual titration, right? So some mornings I need 1 cup of coffee and I'm good for the day. Some mornings I need 3 or 4 just because I've got to have that extra thing there.
Mm-hmm.
So we self-titrate. We self-titrate with the natural product. We have no idea what the chemical profile is of the coffee we drink every morning. And it's very similar to the traditional use in Malaysia of Kratom tea. So they're drinking it and titrating themselves with what's there. So it's really difficult to predict where differences are coming into play. And then on top of that, export material from Southeast Asia that's coming to the United States really has a targeted in range or Mitragynine range that they're looking for a percentage of, and they want that to be the highest alkaloid within the plant. And they've all known that they want to minimize 7-hydroxy and stuff they're exporting. And so we're seeing a much more homogeneous product that's exported, but it is sort of a, if you will, a preferred profile of what the marketplace in the United States is wanting to sell.
Okay. Are there any developing studies to watch right now that you're doing?
Right now, we're involved with the FDA on the human clinical trial, looking at what they call a single ascending dose trial. These are typical trials that are done with pharmaceuticals to start to see where adverse events come into play, or what we call NOELs, no observable adverse event levels. And this starts to give you an idea of what range a substance may be safe to study for efficacy. So the FDA is actually looking into is kratom safe, and at what dosage levels is it safe. I'm not sure that they'll come out and say it's safe, so to speak, but they'll say that there was no, significant adverse events up to a certain level in a single dose in a healthy normal volunteer. So these are not multiple dose studies, they're not chronic dosing studies, so we really don't know how to extrapolate to that yet. But these are exciting because we're actually getting human data. A couple of other trials that are underway that will be published. There's a couple of trials that have already been published, particularly from Mary Payne's lab at Washington State University. They actually published the first human clinical trial in the US in 2022. They followed that up with another study that they did. There's a study that's been published from Malaysia and another one from Thailand. And more recently, just in the past several months, was a publication, a study that was done in Canada with kratom leaf material. And so we're starting human understanding of what's happening. But again, most of the decisions for these trials were made a few years back. And at the time, leaf material was still the predominant market. And now, that's changed completely. And so we're going to start to understand a little bit better what's happening with humans. But the marketplace has changed and evolved so much. These concentrates have become a bigger question. And so we've submitted grants to do a similar thing. If the National Institute on Drug Abuse will fund those grants, we'll be able to study some of these humans and really start to get some better understanding of what's there. But again, that's not going to be done for another few years. And if we get the funding today, it's still 3 years till we have some result in a minimum. And where's the market going to be in 3 years? I don't know. The other thing that I would say is important to watch right now is what's going to happen in regulation. What's going to happen in the state level with kratom consumer protection acts, or the banning of kratom products in certain states or municipalities. And we'll just have to pay close attention to that. And we realize the importance of what it is that we're doing, and all of the finished in science around kratom to get answers to the public and to regulatory bodies, so that we can understand, what is something that we would say would be a limit on, say, a concentrate product. When you get to a certain point, this is dangerous, and dangerous products shouldn't be readily available in the marketplace. How should we be looking at labeling these products to most inform individuals? We have another study going on where we're looking at kratom use in pregnancy, and we're trying to understand, what sort of effects does kratom have on the pregnant mother, and more importantly on the developing child. And so there's a lot of things that are coming in the future. We'll just have to see where things go, and hope that we can get useful information out to benefit people to continue using it, or to understand where we should put the limits on not to use things.
Okay. I know you're not a statesman, but let's say you become Emperor McCurdy Is there any, congratulations, is there any legislation that you would pass right now, if you could executively, about isolates or about extracts?
Oh yeah, my personal feeling, and this is based in me being a pharmacist first, and then being a scientist second, and really having a concern for public safety. I don't think isolates should be in the marketplace. Period. I'm very concerned. I'm also very concerned about this escalation in concentration of X. Um, Essentially, these isolates are almost pharmaceutical products, and they're unregulated pharmaceuticals. But we have no idea what their pharmacological effects are alone, and you know, everything is toxic at a certain level, and so putting things out that are so far manipulated from what nature's provided us is really dangerous. And you can look at it from examples that we already have from natural products, right? Morphine coming from the opium poppy, cocaine coming from coca leaf. Coca leaf is still consumed in traditional manners with no problems in South America by indigenous populations for improvement of work. Yet cocaine, we know, caused major problems worldwide as far as an abused substance. And so, as we start to isolate these compounds out of kratom, I hate to see those be what kills the entire industry for everyone, and suddenly we have no access of any material because it's going to turn into, sadly, coca leaf or opium poppy.
That's really interesting. It seems also like your people in Florida in general is it's really invested in growing a lot of kratom. And are we trying to move toward like self sufficiency here? Is there a goal in mind? Do you know about that or?
There's, I don't know if there's a specific goal in mind. I do know that there are farmers that are very interested in producing, uh, growing Mitragynine speciosa on their farms. In the state of Florida, we have a problem with a disease called citrus greening. It's a virus that's infected the citrus trees, and it's slowly killing the citrus trees. We don't have a cure for it, and we're losing citrus groves in the state of Florida. And one of the things that the state thought would be an alternative was industrial hemp, and that has sort of turned out to be not as lucrative as people had thought originally, because that market exploded and you can pretty much grow hemp everywhere. Mitragynine speciosa is interesting because it needs a tropical environment to survive. It can't handle frosts. So that makes most of the United States unsuitable to grow it in. And Florida has one of the better climates, along with obviously somewhere like Hawaii, Puerto Rico, Guam, that are U.S. territories that certainly could support growth of tropical plants. And yes, there are farming operations in Florida that are producing local kratom for the local markets. They're producing a much more traditional product like you would see in Southeast Asia. And so, as I mentioned, the fresh leaf seems to have no issue in historical use for centuries in Southeast Asia, but we've seen problems with the dried leaf material that comes over here, and there's a definite chemical change in the profile. What exactly that translates to in terms of safety, we don't understand, but there is a desire to have fresh kratom leaf material available in the United States and be able to consume in a manner much more in line with how the traditional usage has been.
All right, and another hypothetical before we close it out. Um, so let's say Gus Fring comes to your house and gives you a bunch of money, and now you have this whole black box project that you can do, and there are no limits. What's a study on kratom you would like to do if you were the designer? Again, you're emperor again, basically, except you're emperor of science now.
Yeah, it's a great question, and I think if you ask me again next week or next month, I would probably have a different answer because that's how fast everything is evolving. But if money was no object and regulation was really no object, we would try to get as much human data as we could with all the different types of products that are out there, including isolates and whatnot. Just because you know some science behind all of these things will really help us understand if there's safety concerns or where there's going to be safety concerns, but at, at what point do we really concern ourselves with those levels? One thing that we are doing, which which is really kind of fun, in our grant, one of our grants that we have, is really looking at understanding what alkaloids are exposed when someone ingests a kratom product, what is exposed into the blood, and then taking those specific alkaloids purified and reconstituting those into a cocktail, and then trying to see if we recreate that same exposure without the rest of the plant material, um, and then see the behavioral pharmacology lines up still. So essentially, is it all alkaloids, or is there other parts of the plant that are really important that we are completely overlooking right now? Because there's many classes of compounds within the plant. Everyone has been focused on alkaloids only. There's many other things like terpenes, tannins and acids and steroids and flavones and just many classes of compounds that we've pretty much ignored up to this point. That will be the one thing that we're really interested in, and then we're going to do what I call leave one out formulations, where we sort of take that ratio of alkaloids that we can gain exposure with, and then take Mitragynine out and see what happens, right? And systematically sort of make combinations so we get every iteration possible, to see what types of therapies we'll be able to develop with those different pharmacologies. So if you will, to go to that orchestra analogy, instead of taking a single instrument out, let's take orchestra and see what it's singing, right? So those are a lot of the things that we're just really interested in doing. I think, though, if I had money today and money was no object, we would definitely try and chase down as much information as we could about products that are emerging and what the effects are on humans.
What a fascinating life you have, and what a fascinating job you have. One more question, I guess, and then I promise it's over. If you couldn't have been a scientist, what would you have become?
Yeah, it's a great question. I mean, as a kid, I wanted to be a professional fisherman or a professional baseball player. My athletic abilities are not that great, so
You can lift weights with your
I
brain. You're good.
yeah, honestly, in as my life has developed, chemistry. I love chemistry. Chemistry is a lot of cooking. It's a very different type of cooking that you don't want to eat, but I love to cook, and so I every night make dinner from scratch. And then I love to pair my. I've become a real foodie. I love to pair dinners with wine, and I've really gotten interested in wine chemistry and viticulture and sort of the whole process of grape growing, and fermentation and wine production. So, I joke around a lot that when I retire, I'm going to become a winemaker, but I think that's like a cliche thing that a lot of people end up doing. And so I don't want to lose the love I have for those things. And so I'm just going to keep doing my day job. So,
And you're doing a great job at your day job.
yeah, sure. I'd love to come back if you want to do a part two sometime. So just let me know. Very good.
Okay, excellent. Thank you very much, Dr. McCurdy. Have a good day.
Yeah.
Good luck at
Yeah.
your event.
Thanks, Lawrence.